In this lecture, we will be discussing about the structure of antibodies. Structure of antibody or immunoglobulins. Okay, antibodies or immunoglobulins. We have already discussed about the innate immunity and acquired immunity. We have discussed that innate immunity is a non-specific type of immunity and that is inborn. And when we were talking about adaptive or acquired immunity, that is specific type of immunity due to the production of these antibodies or immunoglobulins, because their antigen and antibody reaction, they are very much specific. Every specific type of antigen has an every specific type of antibody, or every specific type of antigen has the capability to produce or to make our body to produce that specific type of antibody against it. Okay. So, this was about our two types of immunities that we have already discussed, and these immunoglobulins, these are the part of a second type of immunity, that is acquired, adaptive or specific type. Okay. Now, immunoglobulins, we can also denote it as Ig. Okay. Ig is the abbreviation of our immunoglobulins. Now, we know that these immunoglobulins are generally present in serum part of our blood. Okay. Immunoglobulins are present in serum part of blood. And what is the serum? We know that our blood is made up of two things, that is, one is plasma cells and one is formed elements, and that plasma cells comprises of about 55%. Okay. Our blood is made up of two things, one is plasma cells and one is formed elements. Okay. These plasma cells contains all the protein part, many inorganic ions and blood clotting factors are also present in this. Blood clotting factors, and this formed element comprises of RBC, WBC and platelets. Okay. This is the composition of our blood, and 55% comprises of plasma and 45% comprises of formed elements. Okay. Here in this part, in our plasma part, immunoglobulins are present in plasma part. So, there is a method known, in that method we are taking a tube. That method is known as erythrocytic segmentation rate, ESR, erythrocyte segmentation rate. This is one type of method. In this method, generally blood is taken in a tube. That tube is known as Winthrop tube. Okay, specific for this kind of method. Blood is taken into a tube that is known as Winthrop tube. Okay. Now, we know that the formed elements, they are quite heavy, they are viscous and they are quite heavy type of So after some time this formed elements will be deposited in the lower portion of this tube. They get sedimented, okay. Their sedimentation rate is quite higher, so they get sedimented at the base of this tube, and the upper remaining portion, the upper fluid that remained in this was plasma, okay. This was plasma and this was our formed elements. They were sedimented because their sedimentation rate was quite higher and they were heavier than our plasma material, okay. Now, when this plasma and formed materials are allowed to coagulate, okay, then when the coagulation is to be happen in this plasma, so the liquid which remains behind that liquid is known as serum, okay. When we are allowing this plasma to coagulate, then the liquid that remains behind, that liquid is known as serum, and in that liquid, in that serum, our immunoglobulins are present, our antibodies are present, okay. That's why we said that serum are rich in proteins, serum proteins, that are albumin is one type of serum protein, okay, and that is also present in this serum, and along with that immunoglobulins, that is our antibodies, these are also present into our serum. That's why when we were discussing about artificial passive acquired immunity, so at that time we have discussed that we are taking the blood or we are taking the antibodies from a hyper immunized person. So we were not taking the exact blood of that hyper immunized person, we were taking the serum protein, serum part of its blood. We were taking serum part because our antibodies, immunoglobulins, are maximum in serum part, okay. And one more thing, if we are taking that serum part from any other animal, so we will not say that we are taking the serum, we will say that we are taking the anti-serum, because it is not of our body. If it is self serum, then we will say that it is we are taking the serum, or we are having serum in our blood. But if that serum we are taking from outside, we are taking that serum from any other animal, then we will say that we are taking anti-serum. Likewise, we are taking anti-venom, so we will take anti-serum, okay. Now, immunoglobulins are present in serum part of our blood, and these immunoglobulins are heterodimers. Heterodimers. What is the meaning of this heterodimers? Heterodimers means made of two different types of molecules. The thing which is made up of two different type of molecules, two different type of things, that is known as heterodimers. And here why we have called this immunoglobulin as heterodimers, because it is made up of two amino acid chains, two different types of amino acid chains, okay. We are having two different type of amino acid chains, one is light chain and one is heavy chain. So it is not necessary that the two amino acid chains, that is heavy chain and light chain, will be made up of the same sequence of the amino acid, okay. So that's why different chain, different sequence of the amino acid is present in that chain. That's why we said that Ig or immunoglobulins are heterodimers, made up of two different type of molecules or two different type of macromolecules more exactly, and here the two chains are made up of two different type of amino acids. Now, two different chains, then which are these two different chains? 2 light chains and 2 heavy chains. Okay, 2 light chains and 2 heavy chains. That's why we said that general structure of antibody is made up of 4 polypeptide chain. Okay, this is the reason why we said that general structure of antibody or immunoglobulins is made up of 4 polypeptide chains. Okay, now let us draw that structure. Okay, this is the common structure of our antibody. We are having 2 light chains. These are our light chains. And these are our heavy chain. Okay, 2 heavy chains and 2 light chains comprises of 4 polypeptide chains made up of amino acids. Okay. Now, these are 2 light chains and 2 heavy chains. The structure which is present here, this structure is known as hinge region. This structure or this part is known as hinge region. Okay. Now, why the structure is like this, fork like structure? Why? What is the significance of this structure? The significance of this structure is that it is having 2 antigen binding sites, and one more thing, that these 2 heavy chains, these are joined together by disulfide bond. Okay. The 2 heavy chains are attached to each other by disulfide bonds. Okay. And the same here also, the light chain and heavy chain. Light chain is attached to our heavy chain with the presence of or with the help of disulfide bonds in both the chains. Okay. Now, which amino acid is responsible for this disulfide bridge or for this disulfide bond? There are 2 amino acids, methionine and cysteine. Methionine and cysteine are 2 amino acids which have sulfur group in them, and where these 2 amino acids will be present, they will create the disulfide bond. This is why our light chain always remain intact with our heavy chain, and the portion, the below hinge region, the portion which is present below hinge region, this also remains attached. And here in this portion, there is no disulfide bond, very few disulfide bond, or there is no disulfide bond. That's why these two fork-like structure are being made, and they are separated from each other, widely separated from each other. Okay. Now, other than these, there are many of bonds which are present for the stability of this immunoglobulin. They are, first we should write here this, that light chain remain attached with heavy chain by disulfide bond, and the same with the same bond present between heavy chain also. Okay, light chain attached with the heavy chain with the help of disulfide bond, and two of these heavy chains are also attached with the disulfide bond. Now, some other bonds that which are responsible for stabilizing this immunoglobulin molecule are some other bonds are non-covalent bond, salt bridge, hydrogen bond, and hydrophobic bond. These are other 4 bonds other than disulfide bond which are present in immunoglobulin molecule for maintaining their stability. Okay. And one more important thing, that immunoglobulins are glycoproteinaceous in structure. They are glycoproteinaceous in structure, and the glyco part will be our oligosaccharide. Okay, oligosaccharide, the glyco part, the carbohydrate part. Okay, we are having 3 types of carbohydrates, that is monosaccharide, oligosaccharide and polysaccharide. So, the glyco part here in the immunoglobulin is made up of oligosaccharide. Now, back to our structure, that we have discussed that we are having 2 light chains and 2 heavy chains, and here the upper part, upper 1/3 part we will say, here in upper 1/3 part there are is having 110 amino acid, this upper one part, and this is most variable part in immunoglobulin. Most of the varieties or most of the differences are present in this upper 1/3 part. Okay, that's why this part of the chain is known as variable part or variable sequence, because here this part comprises of 110 amino acid. Okay, now these 110 amino acids will be different in the same immunoglobulin. These will be different, these 110 amino acids will be different, these will be different, and these will be different. So, the sequence of that will be highly varied from each other. Okay, that's why this upper 1/3 part is termed as variable part or variable sequence, okay, and called variable sequence. So, we will give the term variable part of light chain and variable part of heavy chain. So, this one is variable part of light chain, variable part of heavy chain, and this one variable part of heavy chain and variable part of light chain, okay. And one more thing, this terminal is N terminal and this terminal is C terminal. The part near to the N terminal is variable part, okay. Now, the lower portion, that is remaining 2/3 part, here also in both the chains 1/3 part, okay, here also 1/3 part, 1/3 part is variable part and here also 1/3 part will be the variable part, and the remaining 2/3 part, this part, they are having the same sequence, okay, they are having no any kind of difference in the sequence of the amino acids. That's why this part is known as constant region of immunoglobulins, okay. Lower 2/3 part is having same sequence and it is called as constant region of immunoglobulin. So, we will term it as constant region of light chain and constant region of heavy chain. So, here this one is constant region of light chain and this one will be constant region of heavy chain. Constant region of light chain and constant region of heavy chain. That's why due to this phenomenon or due to this arrangement, this immunoglobulin is also called as dimer of the dimer, okay, dimer of the dimer. If dimer means the combination of two things, okay, already there are two light chains, there are two heavy chains, so it is the dimer of the dimer, okay, the term heterodimer also and dimer of the dimer, okay. Now, one more thing, in variable region we have discussed that our upper 1/3 region, which comprises of 110 amino acids, is termed as the variable region of light chain and the variable region of the heavy chain. In this variable region there are some areas are present. In some regions are present, we should say, okay. When variable region, some highly varied or differentiated regions are present and we call it as CDR, that is complementarity determining region. Complementarity determining region, and this specific region is having antigen binding site. Okay, we know that in immunoglobulins, in antibodies, there are specific antigen binding sites. Okay, so where these antigen binding sites will be present? In upper 1/3 part, which is known as the variable region of a particular antibody, and in that variable region also there is one specific region that is known as complementarity determining region, and this complementarity determining region is that specific region where antigen binding sites are present or antigen binding regions are present. Okay. This is the region where antigen binding sites are present, antigen binding sites. Actually, when we magnify this structure, no, so it appears like somewhat bulgy, like this. This kind of structure appears. So this bulgy kind of structure here which is appearing, this is actually our antigen binding site. Okay. When we magnify the structure of antibodies, so some bulgy type of small structures appears in that, and these bulgy type of small structures, these are actually antigen binding sites, because what is the significance is that for binding of any antigen, for any binding, we should have some type of cavity or some type of depression, then only we can, something can hold that foreign antigen. So that's why for holding that foreign antigen strongly in our antibodies, these depressions are present. Okay, and these are only the antigen binding sites. Now, what we have discussed here, that Ig, that is immunoglobulins, are present in the serum part of our blood, and they are called as heterodimers and also dimer of the dimers. Okay. Now, there are 2 light chains and 2 heavy chains in this, and the general structure comprises of 4 polypeptide chains, that is 2 light chains and 2 heavy chains. Light chains remains attached to our heavy chain by disulfide bonds. Okay, and the same with our heavy chains also, 2 heavy chains comprises or combines or attached together with the help of the same disulfide bond, and the reason for the presence of this disulfide bond is our amino acids. We are having 2 amino acids, methionine and cysteine, which are responsible for the production of these these disulfide bonds, because they are sulfur containing amino acids. Okay. Now, there are few more bonds, non-covalent bond, salt bridge, hydrogen bond and hydrophobic bond, which are present to provide the stability to this molecule, to this glycoprotein molecule that we are saying immunoglobulin or antibodies. Now, there are certain variable regions and certain constant regions in this amino acid, in this immunoglobulin. The constant region is the region for antigen, the variable region is the region for antigen binding site. Now, do you get the significance of this antigen, is this variable region, that why variable regions are present in every antibody or every immunoglobulin? Because every antigen is of different type, every antigen has its different nature, its different composition. So, if all the all the immunoglobulins will be having the same structure, same identical chains will be present in all the immunoglobulins. So, how the different antigens are going to detect by these immunoglobulins? Okay, this is the significance of having variable chain or variable part in our in our antibody, that to determine the different types of amino acids, antigens, to determine the different types of antigens, we are having different region, that is known as variable region. Now, we call variable region of light chain and variable region of heavy chain. As same, we are having the constant region, the remaining 2/3 part we call it as constant region, because it is having the same sequence. There is nothing different in this constant region of both the chains. That is why we call it as constant region, and we call constant region for light chain and constant region for heavy chain. And we have also named the N and C terminal, likewise they are present in any polypeptide chain. Okay. Now one more thing, that variable region is having complementarity determining region, and this is the proper specific site for antigen binding. Now, this is our general structure of antibody. Generally, all the antibodies will have this kind of structure. But we have studied that there are 5 types of antibodies, 5 different types of antibodies. So, what are they? What are they formed of? What are they composed of? Here we have taken the one general structure. So, what is the difference between these 5 antibodies or these 5 immunoglobulins that we are having in our body? So, the difference is before that, from where we are getting these 5 types of antibodies, let us first discuss that. Okay. Some scientists were studying over this antibodies, they sequence the heavy chain of these antibodies, okay, and specifically the constant region. Okay, this heavy constant region they study over this part. Now, sequencing of heavy chain. They sequenced this heavy chain and they derived that after that sequencing that this heavy chain differs in all the immunoglobulins. Okay, but we are saying that identical. I will tell you. Sequencing. They sequenced the heavy chain with myeloma protein. They sequenced first this heavy chain. Sequencing means they are just sequencing the amino acids one after one, which amino acid is present and that amino acid is comprises of which type of codon. So, this type of studies is known as sequencing. So, they sequence the heavy chain, they find out that which type of amino acids is present and which type of codon that amino acids, all the amino acids are having certain kind of particular kind of codon. So, what codon that particular amino acid is having. So, this was the sequencing. They done that sequencing by myeloma protein and they reduced it with mercaptoethanol. It is one kind of reducing agent. So, they reduced the sequenced heavy chain with mercaptoethanol, that is one type of reducing agent. And after that, they differentiate the chains by gel electrophoresis. Okay, after sequencing many types of amino acids, many types of immunoglobulins they had taken, and they sequenced one by one with the help of myeloma protein, and after sequencing they reduced that chain and differentiate and under gel electrophoresis by the procedure of gel electrophoresis. So, after the sequencing what they get, there are 5 different types of heavy chains. After sequencing they find out that there are 5 different types of heavy chains present in immunoglobulins, or one immunoglobulin will be having the 2 chains only, but there are many type of immunoglobulins. They have taken the immunoglobulins in bulk. So, while that bulk sequencing they found out that the heavy chains all these antibodies were having, they were of 5 types, and they called it as isotopes. Okay, isotopes. What are these isotopes? These are 5 different type of heavy chain sequencing. 5 different type of heavy chain sequencing is known as isotope. And which are they? These 5 chains, they are alpha, gamma, mu, delta and epsilon. These were the 5 different types of heavy chains that they had sequenced. They had 5 different types of isotypes: alpha, gamma, mu, delta and epsilon. And on the basis of these different types of isotopes, they had isotypes, they had derived the name of each particular antibody. On the basis of these different types of heavy chain, the name of 5 different immunoglobulins is derived. So, what are these names? They had given name alpha. For alpha chain they had given name IgA. For gamma they had given name IgG. For mu they had given name IgM. For delta they had given name IgD. For epsilon they had given name IgE. So, these are the different types of isotypes which the scientists got at the time of sequencing of various kind of amino acids, and on the basis of these different types of heavy chains they had derived these names. Okay. So, likewise we are having 5 different type of antibodies: IgA, IgG, IgM, IgD and IgE. These are our 5 different types of immunoglobulins. Likewise, they had also sequenced our light chains, and they got 2 more isotypes of light chains. They called it as kappa and lambda. Likewise, they also sequenced light chain and got 2 isotypes. These were kappa and lambda. So we are having 2 different type of light chains and 5 different type of heavy chains. Okay, and these will be identical, like we have said that there are 2 heavy chains and there are 2 light chains. So in any of the immunoglobulin, in any of the antibody, the combination will be 2 heavy chains of alpha, 2 isotypes, 2 different heavy chains of alpha, that is here also alpha chain, here also alpha chain, and identical light chain, that here also will be the kappa or lambda or kappa or lambda. Likewise in all the antibodies, either of the chain will be present from this 5, either of the chain will be present and that too identical. If mu chain is present, then this one will be the mu. If here kappa chain is present, then here they will be the kappa. So our antibody is made up of 2 identical light chains and 2 identical heavy chains. That's why we have given that 2 identical heavy chain and 2 identical light chain, because of this combination. Okay. Now one more thing, that in this IgA and IgM, some more variations were observed in IgA and IgM. These were further sequenced to know that whether in this type of antibody there more modifications or more differences are present or not. So when scientists sequenced this IgA, they got 2 para isotopes, 2 more isotypes of this. They named as A1 and A2, or we named this immunoglobulin as IgA1 and IgA2, 2 paratypes of IgA. And likewise when they sequenced IgG, they got 4 para isotypes of IgG. They had given name Ig gamma 1 or IgG1, IgG2, IgG3, IgG4. Likewise, they have sequenced IgG also, and the chain here is of gamma and here the chain is alpha. Okay, we are having 5 different types of antibodies. They are IgA, IgG, IgM, IgD and IgE. Depends upon the type of chain they are having, and after sequencing IgA and IgG, they got the information that these are further having their para isotypes, and they had given the name IgA1, IgA2 and respective to the IgG antibody. Now one more thing, some more experiments were done, or we can say some more tests were done over these antibodies, and one more phenomenon is that these antibodies or these IgGs were made to digest with an enzyme pepin. This is an enzyme named pepin. So they digest, or they try to metabolize this or digest this antibody with the help of the enzyme pepin. So what they got 3 fragments. When they digested this antibody, they got 3 fragments. In this, the 2 fragments were identical, 2 fragments were identical and one was different. They got 3 fragments, 2 were identical and the other one was different. Okay. Now when they observed these two identical fragments, they got that in these two identical fragments antibody binding antigen binding site is present. We have already discussed that antigen binding site is present in CDRs, and this CDR complementarity determining region is the part of these two fragments. Okay. This CDR, this fragment is kind of a bigger one. Okay. This CDR is the part of this fragment. Okay. Now, these two fragments were identical and one was different. So, this was the two identical parts. In this parts, antigen binding region is present or antigen binding sites are present. Therefore, they named this fragment as FAB fragment or fragment with antigen binding. Okay, and the specific site is complementarity determining region in this FAB fragment. Okay, FAB. F stands for the fragment and AB for antigen binding site. And this single segment, that is a fragment, is determined or it's named as FC. Why it is named as FC? Because when they kept this segment in cold storage, so this fragment was crystallized. That's why they named this fragment as fragment crystallized. FC, fragment crystallized. How they known? When they kept this fragment in cold part, or when they kept this fragment in cold region, so that was crystallized. That's why they had given the name as FC, fragment crystallized, and here fragment antigen binding site, because it is having the antigen binding sites inside that, and the specific is complementarity determining region.